Throughout the farms and villages of Uganda, life passes from one generation to the next in a cycle that often feels predictable. Yet, the reason you inherit your mother's eye color or your father's hair texture is the result of a specific biological mechanism, not random chance. Take these local Onkole cattle. When a solid red bull mates with a solid white cow, the result isn't always a calf that matches one parent. Often, the offspring displays a patchwork of both colors. In human families, two parents with dark brown eyes can have a child with bright blue eyes. These physical traits follow a precise, microscopic code. By learning to read that biological blueprint, we can predict exactly how those traits are handed down. To decode it, we look inside the human cell. Within the nucleus, DNA is tightly packed into structures called chromosomes. Humans carry 46 of these, arranged in 23 pairs, one set from each parent. A specific section of this DNA that codes for a certain trait is called a gene. These genes come in alternative forms, known as alleles. For example, a gene controlling height might have one allele for being tall and a different allele for being short. This creates two distinct layers of biology. The invisible genetic makeup you carry is your genotype. The observable physical characteristic those alleles produce, like your actual height, is your phenotype. Because the underlying genotype is a fixed set of pairs, we can use simple mathematics to calculate the physical phenotype of the next generation. Gregor Mendel discovered that some alleles are dominant, expressing their effect even if only one copy is present. Others are recessive, appearing in the phenotype only when the dominant allele is completely absent. Biologists use a Punnett square to calculate probability. Crossing a plant carrying a capital T and lowercase t with a short plant carrying two recessive alleles maps the results. The filled grid shows a clear split. Half the offspring are tall and half are short. When one allele is strictly stronger than another, this rule of dominance allows us to calculate generational ratios with mathematical precision. Nature, however, does not always follow a winner-takes-all system. In many cases, two different alleles are equally strong, and neither one steps aside. This is known as codominance. This square explains our Ankole cattle. Crossing a red bull with a white cow creates a genotype where both alleles express, resulting in a roan calf. The human ABO blood system follows this rule. A and B markers share the stage, producing blood type AB. There is also incomplete dominance, where alleles blends. In some flowers, crossing red and white results in an intermediate pink offspring because neither color fully masks the other. Many of our most vital human traits are the results of these complex allele negotiations rather than simple dominant recessive takeovers. A final layer of complexity depends on the human sex chromosomes, X and Y. Because the Y chromosome is smaller and carries fewer genes, males lack a backup for many genes on their single X chromosome. This makes them more likely to express recessive traits like hemophilia. In this cross, a mother carrying one normal allele and one for hemophilia mates with a normal father. The daughters are either normal or carriers. But in the bottom right quadrant, we see a 25% probability of having a hemophilic son. Understanding where a gene physically sits on a chromosome is just as critical as understanding the gene itself. Mastering these rules of probability allows us to move from observing inheritance to actively directing it through selective breeding. Farmers prioritize genotypes over simple visual markers, identifying which breeding animals carry the precise alleles for high milk yield. This genetic math also protects staple crops. Agronomists calculate specific crosses to breed disease resistance into cassava and matoke, ensuring regional harvests survive local pests. These calculated genetic crosses stabilize the regional food supply and strengthen the economic health of entire communities. By reading the biological blueprint and understanding allele interactions, we no longer leave the quality of our crops and livestock to chance. We become active engineers of our agricultural future.